Hello. In Unit 4, the last section we are going to discuss today, it's called as Assessment of New Antibiotics and a part of Section 5, Standardization of Amino Acids. Myself Prashant Naik, Assistant Professor, Department of Pharmaceutics, NGSM Institute of Pharmaceutical Sciences, Mangalore. So what are the things what we are going to learn? Assessment of new antibiotic, what are the relevant benefits including in traditional HTA, reasons for which additional elements or values for antibodies are required, antibiotics, standardization of amino acids. For assessment of new antibiotics, AMR, antimicrobial resistance, it's called as AMR, occurs when microbes, bacteria, virus, fungi, parasite changes in ways making the medication used for curing the infection ineffective. If new antibiotics are not introduced, the patient will die due to previously treatable infection. However, a major issue is adequate evaluation or the antibiotics, particularly by praying or by healthy technology assessment HTA bodies to the take the account assessment of HTA to take account of AMR antimicrobial resistance and reflect the full benefit provided to the patient and society. The key challenge, clinical trials are designed for establishing non-inferiority, while HTA bodies require demonstration of clinical superiority. HTA bodies do not have a mechanism to evaluate the health benefits of antibiotics and control the rise of antimicrobial resistance. So elements of values relevant to antibiotics, they are given in the table 1 and explained as follows. In table 1, if we can see the relevance and the types of benefit, health gain, we can see over here is the insurance value. Okay, at the same time we will see the other parameters in table 1. So these are the elements and value of elements to antibiotics. So relevant benefits include in traditional HTAR, it includes life extension and quality of life gains. It is expected as a criteria for positive HTA recommendation. Evidence required by HTA bodies is often inaccessible for antibodies, superiority trials. Now, what are unmet needs? It includes the severity of disease and the current availability of alternative treatments. It involves the use of priority pathogen least cost effects offsets. It includes the reduction in cost in other areas as a result of the use of new medicine. It involves the use of modeling studies and or the evidence for clinical trials. Productivity benefits. It involves profits or losses related to various values of patient's time receiving medical care or out of work. It involves the use of modeling studies and evidence from clinical trial observational studies. Other benefits relevance to antibiotics are insurance value. Its value of treatment available in the case of catastrophic patient, healthy events, outbreak of MDR infections, which cannot be controlled by existing last line antibiotics. It is analogous to availability of the fire engine. It is needed to need the precautionary principle. It involves use of modeling studies. Diversity values. It is the selection pressure that is the antibiotics can eliminate susceptible species of bacteria but not other resistant pathogens. Thus, pathogens survive and multiply, making antibiotics ineffective. Involves evidence that reducing selection pressure by withdrawing antibiotics for some time may result in restoration of susceptibility. Involves the use of modeling studies. Diagnostic values. If infection is diagnosed at an early stage, appropriate antibody therapy can be started earlier. Need evidence of test accuracy. Ununiqueness or innovative value. It is a potential value associated with a new or unique mechanism of action. Antibodies and novel mechanism of action may avoid problems of cross contamination or resistance occurring in many existing classes. The discovery of a new mechanism of action of an antibiotic makes it easier to follow on products to enter the market. It needs evidence of a new mechanism of action. Now, what is enablement value? It is an availability of effective antibiotics supported many surgical process and treatment for people having compromised immune system. It involves the use of modeling studies, narrow spectrum or spectrum value. Narrow spectrum antibiotics may be valuable than broad spectrum. It depends on the antibiotics also. Uniqueness or innovative value. Powerful value associated with unique mechanism of action. Antibodies, antibiotics, novel action. Avoid problems of cross resistance. Discovery of new mechanism of action. Needs evidence of new mechanism of action. Now, what is enablement value? Its availability of the effective antibiotics supporting many surgical procedure. It involves the use of modeling studies and spectral value, narrow spectrum, and depends on antibiotic. Non-clinical and microbiological data are important for demonstrating the value of antibiotics. Non-clinical and microbial data for antibiotics can predict their results. Due to the difficulties in conducting clinical trial for antibiotics for MDR pathogen, regulatory accepts these alternative types evidence as a part of approval process in the high unmet need. Antibiotics have benefit go beyond the patient treated when patient is treated with an antibiotic. So antibiotic enable other types of treatment and procedure. Infectious diseases, antibiotics also reduce the risk associated with other type of treatments such as surgery and chemotherapy. Now we will learn about standardization of amino acid. Microbial assay were used for the quantitative amino acid since many years. Quantitation, most reliable, sensitive and specific test. This can be of any type, biological material, however, sometimes they are affected by activators or inhibitors present in the material being synthesized, standardized. Inexpensive method and used for analyzing large batches of samples. Thus, if such microorganisms are cultured using small amount of particular amino acid, limited degree of growth will be observed. Gubiri and Susi introduced a modified version of microbial assay utilizing diffusion in gels in clinical biochemistry lab for screening blood samples having phenylalanine levels. The tests are named as Guthrie test. This bacterial inhibition assay is based on the ability of phenylalanine to counteract the effects of beta-2 phenylalanine metabolite antagonist on the growth of special strains of Bacillus subtilis, which depends on phenylalanine for its growth. The test is carried out on agar layer in which the mixture of suspension of B. Bacillus subtilis, minimum amount of growth nutrient and a fixed amount of phenylalanine is added. Filter paper disc of 4 mm diameter are soaked in blood, placed in agar surface, and the blood soaked discs of phenylalanine standard. Agar plates are incubated overnight at 37 degrees centigrade. Bacterial growth is observed only when phenylalanine concentration in blood discs are sufficient to overcome the effect of B-phenylalanine. Growth is observed in zones around each disc. Next day diameter of the zone is measured and related to phenylalanine concentration. So the reference taken is from N.K. Jain, Manivanand Singh Patel, Pharmaceutical Microbiology, two textbooks, N.K. Jain and Patel, Singh and Manivanand. So today what we have learned is about how to access new antibiotics, what are the various parameters we have studied, and then we have studied standardization of amino acid, how to done and how it has to be performed. That's all for now. Have a nice day. Thank you.